Humans are absolutely fascinated by social behavior, social interaction. We think about our own social interaction. It keeps us busy, it keeps us worried. Social interaction, love, fight are also inspiring art, literature, poetry, change the world with war and all sort of more positive romantic adventure that we love to read or hear about. And so for a scientist, there is something very special about how we recognize each other, or more generally, how animals recognize each other. And one essential question in biology or neuroscience is how animals recognize each other. Why is the recognition of individual within a species occurring? Are there specific dedicated brain areas that are specialized into recognizing other animals, and in particular recognizing an animal of the other sex in order to mate and to reproduce? Specific circuit and neurons dedicated to recognize an animal of the same sex that could be a rival, but also how to recognize our offspring, children or infants or pups in animals. All this recognition is absolutely essential. So in brief, an animal needs to recognize a partner or a rival or a kin. And so this is the type of question that we are very interested in, that have really intrigued scientists for a very long time. And at the earlier part of the 20th century, a lot of ethologists have asked this question. So Nico Tinbergen or Conrad Lorenz, observing animals very carefully and try to decipher the rules of this social recognition. So there is a very abundant literature of this time that is extraordinarily interesting, in which in particular, to give some excerpt, Tinbergen, for example, was observing sticklebacks, which are these little fish that have a very elaborate mating ritual, in which the male has a specific type of behavior that elicit a very specific set of responses from the female. This leads to the female to lay eggs and the male to then take care of the eggs together with the female. So there is really an exchange of signals between both that is extremely stereotyped. And so if it's absolutely identical from a set of individual to the other, then there must be something in the brain that fixed this behavior. The original question, the first question then to understand these behaviors is, what are the signals that elicit specific behavior? How does an animal recognize, oh, this is a female, this is a potential mating partner, or this is a male, I have to fight because that's a rival? And Nico Tinbergen, for example, who was extraordinarily influential and still is, discovered that animals often recognize each other not by recognizing a set of very complex features, but by recognizing what he calls sign stimuli, which are extremely simple features that in essence symbolize an entire stage or sex or gender or physiological status of an animal. And one discovery is led to very famous anecdotes. He had this stickleback fish in his lab, and he had mature males. And males develop a red belly, and that's a sign of a sexually mature male. And he realized, to his surprise, that every day at the same hour of the day, these male fish were all adopting an aggressive posture, which is the head to the bottom and exposing their red belly. Every day, same time. And so he started to look what was happening at this time. And he discovered that every day at the same time, in the road adjacent to his lab was a truck of the mailman that was driving by, and that truck had a red color. And so it made him think that maybe this male fish identified this red truck as another male, and therefore all displayed this aggressive posture. So he tested this by showing to this fish a little decoy of a fish, representing a fish quite exactly. And if that decoy was all very representative of the fish, but without any color, it wouldn't do a thing. But if he took a very simple decoy that was basically just an oval shape decoy, but the bottom half was red, that was sufficient to elicit this very stereotype aggressive posture. And so from this and a lot of other experiments, he realized that all what the fish needed was to have this very simplistic representation of another male fish. And the sign stimulus of aggression was simply this oval shape with a red bottom. There are many, many more examples of this. The way the seagull recognize the newly hatched birds and start to feed them because they get a specific signal. All throughout examples of in fish and in insect and in birds, there are this very interesting recognition of simple signs. Scientists working with mammals try to see what are the specific signals that trigger social behavior. They try to impair vision, didn't do anything, to maternal recognition, for example. They tried to impair olfaction, didn't do anything. They tried to impair touch, it also didn't do anything. But if you impair several of these sensory modalities, then now the mom could not recognize their offspring in the rat, for example, or in other mammalian species. So from this came the really basic question of how do mammalian species or animals in general recognize their offspring or their mate or their rival. How do you analyze behavior? Well, you observe animals, and for a very long time, the way to observe animals was to go to the field and observe animals in their natural habitat. But there are very big limitations to doing this, because many animals go underground, for example, and so a lot of interaction are not visible. And then every person doing a field study will probably have different observation, because the system is not quite identical from one observation to the other. So a big revolution really came in the development of genetics, and a very big name in the field, a pioneer in the study of the neurobiology of behavior, was Seymour Benzer. Seymour Benzer was one of the pioneers in molecular biology, and he hypothesized that one could also study behavior by using genetic methods. And his model of study was Drosophila. And he simply tried to study mutants for specific genes and look at their impact on behavior. It was a huge bet, because many scientists at the time were saying, oh, behavior is much too complicated to be reduced to the function of genes. And I guess he had this intuition of the genetic basis of behavior from looking at simpler organisms such as viruses or bacteria. If you affect a gene, if you mutate a gene and it affects the behavior of a bacteria, why it wouldn't affect also the behavior of a Drosophila, for example, or the species? And so his studies, his experiments have been really, have revolutionized the field of behavior study or neuroscience of behavior. He isolated mutants of the circadian clock. He isolated a very famous mutant called fruitless, which is a mutant of sexual behavior. He identified mutant of memory, learning and memory. And so these were really the first examples of mutants, meaning a specific gene being affected and non-functional, being related to such a complex function as a behavior. And so from Drosophila, then other scientists got inspired to look at mice or a rat or other animal models. And one of the Drosophila is still a fantastic model system to look at behavior. Other invertebrate systems, such as C. elegans also, and they are extraordinary scientists right now working on these animal models. In mammals, the model of choice is the mouse. It's a system in which one can abolish the function of specific genes using genetic recombination or genetic engineering in general. And so the idea is to try to identify what are the key genes that affect specific behavior in the mouse. And we are particularly interested in social recognition, as I mentioned. And in the mouse, or rodents in general, the way animals recognize other mice is through the sense of smell. And so the olfactory system is really essential for the animal to be able to recognize its surround and to be able to recognize potential mates, rivals, kins, etc. The field of olfaction really was entirely revolutionized by the discovery in 1991 of the olfactory receptor gene by Linda Buck and Richard Axel. So they discovered that a very large family of 1,000 genes encode a very diverse family of receptors that are specifically expressed in the nose. And each receptor is able to detect or recognize a very specific set of molecules of odorants. In rodents, there is also another set of olfactory neurons that are specialized into detecting pheromones. When I was a postdoctoral fellow in Richard Axel's lab and then in my own laboratory, I discovered the receptors for pheromones, receptors that are expressed by a set of neurons located in a part of the nose called the voronasal organ, or VNO for short. And so what we discovered is that the VNO receptors are completely unrelated to the odorant receptors. So the idea is that there is something very special about the molecules that convey social information. So pheromones are, by definition, molecules that are used by animal, released by animal to provide information about their sex or their physiological status or their genetic status. And so with the discovery of these VNO receptors, now we could address very specific questions about how are these social cues identified and what do they do to the brain. And we were able recently to pair specific receptor molecules to a specific source of information. And to our surprise, we actually discovered that many of these receptors are involved in detecting predators. So this set of neurons in the voronasal organ are not only essential to detect a female mouse or male mouse or an infant mouse, but also a very large category of predators. Some receptors are specialized into detecting mammalian predators very specifically. Some are specialized into detecting birds of prey or reptile. Some receptors are even more specialized to detect one particular mammalian predator, let's say fox or particular type of snake. And so our work now in the future is to try to understand how is the specific information for a particular predator or a particular mouse, type of mouse, then relate to the brain. How does the brain make sense of these various signals? So the neurobiology of behavior is, I think, is interesting in many ways, but I think there are two very compelling reasons. One is the brain is here to organize behavior. That's the function of this organ. It's an extremely complex function, and we don't know anything about it. And so the hope is to understand the signaling throughout the brain. How does the brain decipher signals from the outside, computes them, and then comes up with a behavior. This is what the brain does, and addressing this question very specifically would give us insight on what is the biggest black box in our organism. That's the brain. We don't really know how the brain works, and this is a way to directly approach that function. The second very compelling reason is the fact that because in part we don't understand the brain, we also don't know how to diagnose mental illnesses, and we don't know how to cure or treat mental illnesses very well. If we understand some basic mechanism of how behavior is generated, then we will hopefully be able to have better diagnosis for disease like autism or schizophrenia or depression, where patients are really impaired in their social recognition and therefore in their overall behavior in society.